Good morning, everyone. In my presentation, today I am going to talk about correlation between naturally occurring feed mycotoxins and necrotic enteritis severity in broiler birds experimentally induced with necrotic enteritis. So what are mycotoxins? Mycotoxins are basically secondary metabolites produced by fungi on the crop. Mycotoxin contamination could happen either in the field or during handling, during storage. Even though we have more than 200 species of fungi that produce mycotoxins, majority of the mycotoxins family belongs to Aspergillus, Fusarium or Penicillium. We can broadly categorize these major mycotoxins into field mycotoxins and storage mycotoxins. Trichotheceans, fumonisin, zearalenone will fall under field mycotoxins. Aflatoxin, acrotoxin fall under storage category. The biggest factor that determines that amount of mycotoxins are temperature and humidity. These are the major mycotoxins on the left-hand column. That all the major mycotoxins present on that corn, wheat, and barley. Corn, wheat, and barley are the major commodities in the poultry feed. Because of these, mycotoxins occur more frequently in the common poultry commodities, including finished feed, which contains corn, wheat, and soybean meal. Mycotoxins are the biggest problem to the poultry industry. So what is the consequence of ingestion of mycotoxins contaminated diet in the chicken? In the poultry industry, our primary goal is to increase the production performance. Production performance is entirely dependent on nutrition, immunity, and gut microbiome. If any one of these three components is compromised, the other two will be adversely affected. So for example, healthy intestinal tract is very critical for a production standpoint as well as welfare standpoint. Intestinal epithelial cells are rapidly dividing cells with high protein turnover. Mycotoxins targeting these type of cells and inhibit the protein synthesis. So hence, the intestinal epithelial cells compromise several key functions like decreased surface area and available for nutrient absorption, modulation of the nutrient transporters, loss of barrier function. In addition, mycotoxins facilitate persistence of intestinal pathogens and cause intestinal inflammation, ultimately leading to decreased production performance. The intestinal mucosa act as a barrier, preventing the entry of foreign antigens, including toxins and pathogens, into the host. Mycotoxins disrupt the intestinal epithelial barrier, and these disrupted intestinal cell component and undigested food particles can be used as a growth substrate for pathogen like Clostridium. Hence, mycotoxins act as a gate openers, favoring pathogen colonization within the gut. Here I am going to switch my gear towards necrotic enteritis. Necrotic enteritis is among the top disease issues in the poultry, and it causes billions of dollars annual loss globally. The major inducing factor is the toxins produced by Clostridium perfringens in the poultry gut. Because of any factor that can cause damage to the poultry gut, act as a predisposing factor to induce necrotic enteritis. So it is possible that mycotoxins can induce necrotic enteritis. So our objective was to evaluate the role of naturally occurring mycotoxins on development of necrotic enteritis in broilers. So just to be clear that this study was not designed from scratch to study about the role of mycotoxins during necrotic enteritis, but rather we analyzed the data from a big study conducted for different purpose. We analyzed the data from the total of 6 studies conducted over a period of 9 months. We observed that the necrotic enteritis score was different between the birds in the positive control, even though all the trials were run under same conditions. In all the trials, birds were raised in cages with 8 replications in all studies. Male COP 500 birds were used in all 6 trials. On day 0, number of birds placed per pen varied from 12 to 15 depending on the study. We analyzed the data only from the positive control group in those studies for the presentation. So all the trials had the same feed formulation, and they were corn soybean based formulation. The only difference between the 6 trials was that feed ingredients were purchased from the same vendor but from different batches. We sent the starter feed to Romer lab for quantifying feed for these mycotoxins. We did not analyze the finisher feed because the necrotic enteritis scoring occurred at 21 days. The same necrotic enteritis disease model was used in all the 6 trials. On day 14, birds were gouged with Amelia maxima, 5000 oocysts. On day 19, 20, 21, birds were gouged with 10 to the 8 CFU Clostridium perfringens strain NB6. And birds were scored for lesions on day 22 by the same person in all the 6 trials. We measured the gut integrity using the FITC dextran study, where we gouged 2.2 milligram of FITC dextran per bird on day 21. 2 hours later, we measured the FITC dextran content of the serum using fluorescent plate reader. Data was analyzed. JMP disease scores were analyzed using Kruskel-Wallis non-parametric test. One-way ANOVA was conducted to analyze the gut integrity data. Pearson correlation coefficient was calculated between mycotoxins, disease score, and gut integrity data. 3 of the 6 feed had aflatoxin, and 1 of the feed has 16.7 parts per billion of aflatoxin B1. Almost all the feed had fumonisin. 2 of the feed has DON. 3 of the feed had zearalenone. 1 of the feed had both fumonisin and DON. I have organized the data in a slightly better way in the next slide. This is the data from the previous slide. 3 of the feed had aflatoxin B1. For your reference, FDA guidance for aflatoxin in poultry diet is 20 parts per billion. All feed had some amount of fumonisin. For your reference, FDA guidance for fumonisin in poultry diet is 50 parts per million. Similarly, FDA guidance for DON in chicken diet is 5 parts per million. One of the caveat I need to point out is almost all feed had some amount of mycotoxins, and because of this we don't have a real negative control. This means that negative correlation between mycotoxins and necrotic enteritis score needs to be taken with a pinch of salt. I am presenting the necrotic enteritis disease score in 2 different ways. Experiment 1 had 80% of the birds with a lesion score of 2 or more. Experiment 2 had 32% of the birds with lesion score of 2 or more. In all rest of the experiments, less than 20% of the birds had a disease score of 2 or more. So the experiment 1 and 2 had moderately severe disease score. Then the rest, mortality was around 12% in birds. The mortality rate was slightly higher in experiment 1 but was not statistically different from any of the other experiments. Because necrotic enteritis score was non-parametric test, I am presenting the rank sum of disease score. Birds 1 in experiment 1 had a higher disease score than the rest. Birds in experiment 2 had a significantly higher necrotic enteritis score than experiment 5. Similarly, birds in experiment 1 had a higher damage to the gut compared to those in the rest of the experiments. To summarize, birds in the experiment 1 had a higher disease score and had increased damage to the gut epithelium. The aflatoxin B1 content of the experiment 1 feed has 16 parts per billion, and the total aflatoxin content was 20 parts per billion. There was a significant correlation coefficient of 0.48 between the total aflatoxin content and NE score, and also 0.67 correlation coefficient between the total aflatoxin content and gut integrity. There were no or even negative correlation between these three mycotoxins and both NE score and gut integrity. But keep in mind, almost all feeds have some amount of mycotoxins. And because of this, we don't have a real negative control. This means that any negative correlation between mycotoxins and the measured parameters is not real. These mycotoxins will increase the baseline that no meaningful interpretation can be made on negative correlation. Of course, this experiment has several confounding factors. This experiment was run with varied weather. It's possible that Georgia summer had effect on NE score. We used different batches of birds, Emeria maxima and Clostridium. Again, all trials were the part of the bigger trial. In conclusion, mycotoxins present in all poultry feed, albeit at low levels. NE score and gut integrity differ significantly between trials. Total aflatoxin have positive correlation with the NE disease score and gut integrity indices. So aflatoxin above 20.1 parts per billion is most likely involved in increasing NE severity. With that, I would like to thank Dr. Applegate, UGA, Gabriel, postdoc, Kayla is a graduate student for their support during the sample collection, and of course, Romer Lab for analyzing the feed. With that, I will take questions.